All right, hi everybody. Can you guys hear me? Okay. All right, great. So welcome to our ISCC-PEG annual meeting. It's very exciting. This is the first we've been back in person. And we appreciate people who come in person, but also we have about 40 people online, and we're great you could join us that way as well. And we're hoping, you know, we're always excited to have this and to get participation. So just to the people in the room, if you have a question or you want to talk or whatever, or, you know, bring up something, you have to hit the microphone because otherwise the people on Zoom won't be able to hear you, okay? So, and we just learned this mic is always on, so if you have personal things to talk about, don't talk about them over here. So, all right, so we're just going to do a little bit of an introduction. I'm Rich Haspel. I'm at Beth Israel Deaconess Medical Center. And I'm one of the co-chairs, and Donna is our other co-chair from NHGRI. And just a little bit, first of all, it's really thank you guys for being part of this, because if you weren't part of it, we wouldn't have ISCC-PEG. And also NHGRI provides an incredible amount of support. Roseanne Wise, really, is she?
She's getting our keynote speaker.
Oh, she's getting our keynote speaker, but she's been really incredible help. Tasha Stewart, some of you have worked with for travel and things like that, and Dr. Brody, who kind of runs the show in some ways. So we couldn't do it without NHGRI, without you guys. There is a Google Drive with a bunch of info. Sometimes I try to click on the link in the talk and it doesn't work. It's some weird thing about PowerPoint, but anyway, we can get you the link if you don't have it. So, and you know, we are recording everything except the small breakout groups, and all our slides will be available. Okay, so sorry if I missed anyone, but this is probably doesn't even include everybody, and I apologize, but this is just the logos of a bunch of different groups that are part of ISCC-PEG, and I think it's pretty awesome. We have individual members, we have people representing organizations, societies, NIH institutes, we have industry members, and really that's what ISCC-PEG is about, is bringing people together. So, and we usually like to welcome some of our new members. We currently have, give or take, 286 members, 58 organizations, 7 government institute represented, 7 companies, and 138 individual members. We have 31 new members since our last in-person meeting, and we have 16 individual members. We have 11 new people from government and institutes, decent chunk, which is great, are from NHGRI. We also have a new individual from NCBI and HRSA. And we have some new reps from organizations, some of them new organizations, and some of them, your organization been with us, but a new person representing. So first, again, I'm sorry if I missed anyone. Feel free to reach out. I will correct it. It's hard to put all those logos together and things. So, but if we missed anyone in this, please let us know. And please spread the word. We're always looking for new members. It's the diversity. It's people from different backgrounds. You know, I'm a pathologist, so I go to pathology meetings, so I work with pathologists. It's really important to have things like this where we can cross disciplines, we can cross areas of expertise and really work together, because genetics is something where you really need a team approach. So please let your friends and colleagues know about us if they're interested in healthcare provider education. So we're now in our 3rd year of the Scholars Program. I think that was born actually out of one of these meetings, so we welcome new ideas. Ava and Molly are 2 of our new, are in this latest class of scholars. We actually graduated our first class of 4 scholars in 2022, and you can access their presentations. Is that on the main website? Our main, right. So if you're interested in the cool things they were doing. We currently have 6 scholars. We have 2 new this year, as you can see. And just a brief, they're paired with an ISCC-PEG member as a mentor to work on a project. They'll be with us for 2 years, although we definitely, you get free membership afterwards if you want, like everyone else. But no, obviously we hope that people will continue with us, but the formal period of scholar, being a scholar, is 2 years. And in addition, we also provide travel to this meeting as part of one of the aspects. And really, we couldn't also do the scholar program with all the people who have volunteered to be mentors. So really, thank you. Some of the stuff that people have done has been really very cool. So thank you to all our mentors. All right, so just goal of ISCC-PEG, sort of in this whole space of collaboration. We want to determine needs, create useful resources, and we also want to disseminate and learn about best practices. And this is one of my favorite slides. It's from a demotivation slide. Committees, just like teamwork, only without the work. So that's one of the reasons we call them project groups and not work groups. It's a little hokey, but the idea is we want people to have projects, people not just to complain, that's good too. It's good to, and to discuss, but also to create resources because again, we're bringing people together of so many different levels of expertise. So I'm going to turn it over now to Donna to talk about some of the great stuff our project groups have been doing. And we've been getting more and more project groups, which is great. And if you have an idea, please come let us know.
Thank you, Rich. Yeah, the project groups have really expanded over the past couple of years. We have 10. So I'm not trying to be comprehensive as I give this overview of what the project groups have been doing, but a few of the highlights I'll talk about. So our inclusion in the Practice of Genomic Medicine project group, exploring the impact of implicit bias toward disability. They gave a presentation at the Society of Teachers of Family Medicine, had a couple publications, and importantly, one of their first products, speaking of products, was the clinical decision-making in the prenatal setting resource, part of a research study that is also an educational resource. So pharmacogenomics, we have a couple of their folks in the room now and many on the phone. They had several presentations. In fact, I believe Bob Ostrander just walked in the room. So he's here, gave a presentation at the American Academy of Family Physicians, FMX experience. They have a number of resources that I'm told will be out probably in the next month. So we're quite excited about a whole series of resources coming out in collaboration with the University of Pittsburgh, and they will have CMEs, CNEs, physician assistants. Several disciplines can get CE credits through the University of Pittsburgh. So we're very excited. And they will be linked through the ISCC-PEG website. So we do what we can to connect people to the resources that are coming out of ISCC-PEG. The direct-to-consumer genetic testing group, I asked the different groups to give me their accomplishments over the year, and there were so many I couldn't fit them on the slide. So, and that's true for most of the groups. Here's a couple of their presentations. They had a plenary session at ACMG. I was actually a part of that as well. And several other presentations. They had several posters. They're working on a publication. And then the resource that is posted on genome.gov is listed here. That's an FAQ. And the different project groups are also working on additional FAQs. So I am the liaison to our communications team. So that means that we can connect with the communications group to disseminate these resources. The Rare Disease Project Group, I know Rich has been quite involved, and we have Sabrina here in the room. Thank you, Sabrina. Thank you to all of you who are co-chairs for these groups, because this is volunteer work that leads to very important things, but your contributions are greatly appreciated. And so the Rare Disease Group had an iSong presentation in November 2022, and I know Rich was also involved in that. The faculty, we see a few names. We certainly recognize several people who are in the room, including Sabrina, Rich, Rebecca's here too. So thank you. And a comment, we included here, really excellent and engaging. Also, an American Academy of Physician Assistants presentation through the PAs in that group. And a publication. KP is one of this year's presenters of ISCC-PEG Scholars. And she has a publication that she and I believe Sabrina is a co-author on here in the et al. Group. But anyway, so she's doing great stuff too. The OBGYN Genetics Curriculum Group, Barbara and Louise are on virtually, I believe, and they had an abstract accepted, and both Barbara and Louise are mentors to our ISCC-PEG, to different ISCC-PEG scholars, so we really appreciate that. The Adult Cardiovascular Genomics Project Group, we have Siddharth here in the room. Thank you, Siddharth. And his UT Health Cardiovascular Genomics Certificate was actually funded through an NHGRI grant, the training modules in genomic medicine for healthcare professionals. And this is live and looking for other participants. The LGBTQIA+ has been getting together first with the outgoing co-chairs, Kellen Baker and Lila Jamal. And I just learned this week that they have new co-chairs for this group, Kelsey Bonard and Lee Williams, who I believe are on the call. And a couple of their plans are to develop an information page for the NHGRI website to provide appropriate terminology and other additional use of information on the LGBTQIA+ issues in genomics. And I just heard the other day at our council meeting that they're working on collaborations with one of the NHGRI working groups, the Community Engagement in Genomics Working Group. So thank you. We also have Trina in the room representing the Nursing Genomics Group, and they are working steadily on another FAQ, the frequently asked questions about nursing genomics. And those FAQs will address at least 3 different audiences: the entry-level nurses and the general public, advanced practice nurses, and academic nurse educators, as well as advanced practice nurses who are nurse practitioners. So we're looking forward to that as it's developing. And each of these project groups will be meeting, as you know, from 1:30 to 3:30 today to dig into their projects and make further progress. So I'm the chair, for lack of a better term, of Genome Ed, which I think many of you in ISCC-PEG have learned about. This is built on the foundation of G2C2 and is a repository of genomic education resources. So certainly for all of you who are building resources, they can certainly be submitted to that Genome Ed website submission portal. And we're very glad to announce that we have a full cohort for the Genome Ed Review Committee that are listed here. All of these folks are ISCC-PEG members, and again, donated their time to review resources. We have several in the room. And many of these folks work on project groups as well. So they lend their expertise in evaluating and reviewing new resources for this really important repository of genomics educational resources for healthcare professionals. And that has been highlighted through the Genome Ed website and through NHGRI. Another project group that has been in existence long before ISCC-PEG began is G3C, the Global Genetics and Genomics Community, which was begun by Kathy Calzone and Jean Jenkins, true diehards in genomic education. So this is a video-based, case-based program in genomic education. And if I can click on this link, just yesterday, the group released as a online resource on autism spectrum disorder and vaccines. So let's see if I can open that up. Great, okay. So the group has been working very hard on getting this new resource out, Ethan and Di, which is actually a continuation of.
So the other day, I was talking to some of the moms in the.
All right, I think you get the idea. This is a video of a mother speaking about her child who she believes has some interesting behavioral conditions. And vaccines were also part of the concern of this mother, that vaccines might have caused some of those behavioral differences. And so this is a really unique resource in that you can see what the patient would actually be saying and doing. And as a user, you're answering questions that come up with this particular patient. And as you could see in the last screen capture, we have 17 or more cases using this platform. So now,
ISCC-PEG has contributed.
Oh yeah, let's be sure to note that. Several years ago, we had another case come out, and we had ISCC-PEG people reviewing the case and contributing. Some time ago, we sent out a survey to determine the different cases that ISCC-PEG members were interested in. And autism spectrum disorder came out very high on our list of our survey. Okay, so let's see if I can return.
We go down to the PowerPoint. Right there. The one on the right.
There we go. Thank you. Thank you. Okay, and in the talk here, you see I have links to all the cases, to the flyer that provides additional information. So, and I could, each one of you could spend time on your different resources. This is a particular one that's had a lot of history with our branch.
Donna, are you open for questions, or do you want us to wait for the end?
Yeah, let's wait till the end of the talk, but thank you.
Sure.
Yes, you can save your questions.
No, I'll wait for them. Thanks.
Yeah, if you could send it in the QA or the chat, then I can take your questions then. So I think Rich referred to project group versus a working group. We are active in our groups and think about questions like dissemination, where will the resource live? We present at different organizational meetings, and certainly, and you started to see this in my overview, we're getting publications from these groups, which is excellent. These are a couple of things to keep in mind: the conflict of interest, the review and dissemination policies. For any who have not yet seen, we have a policy document that we follow for ISCC-PEG members. And we also have had excellent participation from professional societies, which assist in convening and connecting to the professional meetings. So that's a really important part of this work. As Rich mentioned, we're bringing people from many different organizations together so we can share the genomic educational resources. Every one of the project groups that I've already mentioned has expressed an interest in participating in another year of the Healthcare Professionals Genomics Education Week, which this coming spring will be our third year. And this is a social media event that NHGRI has supported these 3 years. All members and organizations can participate. Typically, the organization hosts an event, a webinar or other event. We've had Twitter chats. We've had a lot of dissemination through social media. And please consider that. And it's coming up in June. We've also supported through our Zoom team some of the projects. And we actually put the word out to all of you members to submit proposals, and that period is finished now. But that's not to say you can't have your own organization host all kinds of events or webinars yourself. We're in the process of evaluating the proposals that were submitted, and that's just another level of support that we're providing. But please remember, you can always host your own webinar and panel, and it will be listed on the Genomics Education Week website. Okay, turning it over to Britt.
Thanks, Donna. Just a word about the compendium. We have, it's a list of, well, it's a booklet of the different organizations, and also the individuals can put a page in to describe their interests and resources they've developed and things like that. So it's just another way to network. And after this meeting, we put out a call for some new entries, and we'll do that a little bit more after this meeting. But once a year, we try to update it so people can just, if they're interested in what other people are doing, or if you want to look for different resources, it's just a place to go. And what's nice is now we not only have the organizations and the institutes, but also individuals can talk about things they're doing. And we also use it as a way to get an idea from you guys of what you'd like to see from ISCC-PEG. This is just a slide that's available on the website. Please, you know, if you're giving a talk somewhere and you have a room for an extra slide, or if you could show something like this, we just really want to get more membership and reach out. And we welcome also, if you don't want to, you know, if you have an idea but you don't know people there, just let us know of a group. We can reach out to them, because again, really the diversity of our membership is really critical. And that's why we put together this slide, to help spread the word if you're giving a talk or something. Okay, so here's our agenda. We're doing the introduction, and then we're fortunate enough, Dr. Green will be here to introduce to give a little update on some NHGRI things, but also introduce our keynote speaker, Dr. Denny, which we're really excited about because of his involvement with all of us, which is an incredible project. And then we're going to have our ISCC-PEG scholars give their presentations. They do this in their second year at our meeting. They talk about the things they've been working on. Then we'll have a little bit, our extramural folks will come in, because again, part of scholarship is looking for grants, and now there are more grants available in education. So to talk a little bit about that, or if you have other questions. Then you can go grab some lunch. We'll have our breakout rooms, and again, people will be able to participate remotely or in person. And then we'll come back, and each project group will just give a brief summary of, very brief, what they're doing, and more about what asks they might have of membership, like what we can help them with as the larger group. And then we'll have a little bit just talk about, you know, wrap up future directions. And please, during the day, feel free to, if you're in the chat, you know, if you want to put up suggestions of things, like I mentioned, the Scholars Program came from a suggestion. We might set up, maybe we could get some sticky notes where people could put up,
Or
you know,
write on it, yeah.
or yeah, even write. If people just, you don't have to, if you want to keep it anonymous, you don't have to put your name, but if you want to write suggestions up there on one of those seats, that would be great, because we're always looking for new ideas. So I think there might be, I know might be one, Gerald, can you look at the chat, or who's best to do that? I don't know if I can, because I think there was a question.
Yeah, there was one question.
It was Maya, but I can wait with it later on. I think we might have a conversation about it also in our small group. So maybe we can report on it later on. No worries. Thanks.
Okay, sure. And again, just because I know, and I appreciate Maya, you even asking the question, because it made me remember, just for logistics. Probably if people remotely can put it in the chat, and we'll check in we'll check in for that, just because otherwise we could have like a bunch of people speaking, which might get complicated. It's one of the challenges of the brave new world with remote stuff. So But anyway, I think we're ready to move on.
I think
then.
we're ready, yeah.
All right, well, thank you again all for coming. I think we have a great program and a great day where we can get some work done too. So thanks a lot.
So I don't think Dr. Green needs any introduction, but we're pleased to have Dr. Eric Green, our NHGRI director, provide NHGRI highlights. Thank you.
Thank you, Donna. Good morning, everyone. Delighted to be here. Let me say a number of things to start. First of all, it's obviously great to be able to do this in person. I hope all of you agree with that. But unlike bringing you to Bethesda and putting you in a windowless conference room at some hotel, we got you a better view and a nicer room. Actually, I didn't know which room up on 6th floor we were until I got up off the elevator to see where it was. But you should realize this is a very well-used room. I'm here almost every week. This is actually nowadays where the NIH Institute directors meet, literally this room, not even the one on the other side. This is literally the room that the NIH director convenes all the institute directors. So, and we've been doing that since we came back in person. We do it hybrid, but we come back in person. And actually, Rich is sitting right where Larry Tabak normally sits. And Larry Tabak is performing the duties of the NIH director. You can't call him the acting NIH director anymore, but that's for complicated legal reasons that is really silly. But and where Larry Brody's sitting is where Tara Schwetz, who's the acting deputy director, usually sits. And somewhere near that side of the table, near the front, is usually where I sit. So in any case, that's that. I am here and was given a half hour, which I don't know if I'll need necessarily to sort of give highlights. This was really hard to sort of pick what to talk about. And so I would strongly encourage you to either interrupt me with questions, or if I don't get to things you want to ask about, ask me. And I'm happy to talk about absolutely anything, but it was a little hard to gauge exactly what would be the most useful. I also feel in many ways that I'm just the warm-up act for Josh Denny, who will follow me, because Josh is just a rising star here at NIH, and I think the world of him, and I'm going to be delighted to introduce him, and he'll give you even a far more substantive talk than I will. And I'm also not afraid of your questions, because I feel like if you ask me anything really hard, I'll just make Larry Brody answer the question. So as long as Larry's here, I feel comfortable. So in any case, what are some of the highlights that might be of interest? And I was reminded that I did talk to this group virtually, because everything was virtual then when our newest strategic vision came up. So I'm going to do it within that context of thinking about of you being at least familiar with the 2020 strategic vision we published. I can't help but start by pointing out, Gerald, is there a way to minimize the thing at the top so they can see my titles? I know there is, because you've done this for me before. There we go. Thank you. 2023. This is a great year, way cool year, lots of things to be excited about. And for those who don't fully keep track of these odometer moments, let me remind you, this is the 70th anniversary of the discovery of DNA's double helical structure. And in fact, I just spent an hour on camera being interviewed by Kevin Davies, who is editor of Genetic Engineering News, for a major video program they're going to be putting out in a couple of months, or maybe like next month, commemorating the 70th anniversary of this incredible discovery that in many ways sort of launched everything. But also shown on the far right is an acknowledgement that April of this year will be the 20th anniversary of the completion of the Human Genome Project, which I don't know about the rest of you, but it gives me pause of, oh my gosh, I can't believe it's been 20 years since the project ended, but April 2023. And that, in fact, aligns with the fact that the 20th anniversary of National DNA Day on April 25th will be celebrated. And again, National DNA Day, congressional proclamation or whatever, it wasn't a law, but it was basically named as April 25th of every year as National DNA Day. And the very first one was 20 years ago to commemorate, oh, there's Josh, to commemorate the completion of the Human Genome Project then 20 years ago. And then 20 years ago was the 50th anniversary of the discovery of DNA's double helical structure. So in any case, everybody should celebrate. Think of the ways you're going to celebrate. Trust me, my communications team and I are working hard to come up with all sorts of celebratory events and commemorations and information dissemination and so forth. So while we're in a celebratory mode, let me see how I advance this. Daryl, can we, again, is that going to keep popping down, or is? I can't help but just sort of set a context in a celebratory kind of mode. Just let me remind you, why are we so excited about celebrating so many things in this first 3+ decades of genomics? The field, I always would contend, is about 36 years old now, with the word genomics being coined in 1987. I mean, just think back. And I actually, looking around the table, some of you were too young to think back. So just join me wherever along the highlights that you came into life. But I get to think back on all these things, and I'm taking Brody with me because I know he's roughly my age. We think back on these things, and we did it. We sequenced that human genome for the very first time as part of the Human Genome Project. We recognized when that was completed that was wonderful, but it was too expensive if we were going to really eventually see this be realized in many nooks and crannies of biomedical research, particularly as part of clinical care. So having checked that off our list, we moved forward and developed all these incredible technologies that have reduced the cost of sequencing the human genome by more than 1 million-fold. And that was a tour de force technological set of advances, both from the public sector and government funding, but also the private sector. And that gave us the tools that we needed to then go out and not sequence just one genome or even a few genomes, but now sequence literally millions of genomes. And with that, we got great insights about the variation that occurs in each of our genomes, and increasingly better views of the variation that occurs in different subpopulations of individuals around the world. But of course, we don't want to just know about the variants. We want to know how those variants influence function. And eventually, especially those of us who think about medical applications, we want to know which of those variants are relevant for medicine and for maintaining health and preventing disease and so forth. But to do that, you need to know how the human genome works. And while we certainly are far from completing that task, I think we can celebrate 20 years of increasing knowledge about the human genome function. And increasingly, we're getting insights about how variation affects function, although, boy, we have a lot more to learn, and major programs in NHGRI and elsewhere are really tackling that challenge as well. But we got enough progress in sequencing capabilities and understanding variation, understanding genome function to really see significant advances on unraveling the genomic basis of human disease, especially over the last 10 years. And the greatest celebration comes with rare diseases, which, as all of you appreciate, is disease that at least you're dealing almost in all cases with a single gene, and so you don't have to tackle the complexities of multiple genes and multiple variants and more straightforward. For those who don't keep track of these numbers the way I like to, I just want to remind you, it's an easy thing to remember for me, that when the Genome Project began, 61 rare diseases for which we knew the gene that was mutated, just 61. And now we're just about to pass the 6,000 mark of having 6,000 rare diseases that we now know which gene is implicated. That deserves a check mark, certainly, although then we have to immediately remind ourselves, but then there's all the common diseases, which are really the things that fills hospitals and clinics around the world. And those are complicated. It's not a single gene in most cases. It's often with a greater influence of environmental and social factors. Harder to tease out. I think we're getting to the point where we'll make great strides in the next decade, but certainly certainly it is a challenge, and that one of the grand challenges we will face going forward. But we've gotten enough progress in those arenas. I know this group is very interested to be able to put up this highlight and celebrate the fact that where once upon a time the idea of genomic medicine was a blurry concept. What was it really going to look like? We now know, at least from some of the earliest examples. They may just be illustrative examples, but at least we can point to specific examples where genomics is being used as part of clinical care. Amongst those examples, I'm often asked, what are the ones I like to name? What do I think are the highlights of genomic medicine implementation? Clearly, cancer, I think all of you appreciate. I think it's going to always probably be the greatest impact in any clinical area, and a lot is happening, and I applaud what's going on at the National Cancer Institute in terms of their significant investments in cancer genomics and their including in very clinically oriented fashion, many trials going on to really figure out what is the best use of genomics as part of oncology. But what I'm also been remarkably impressed with is rare genetic disease diagnostics. I don't know how Rich feels about this, but when I was in clinical training and I first got involved in genomics, I wasn't really convinced that I would see in my lifetime a change in diagnostic medicine that would involve sequencing patients' genomes. Of course, when I got involved in it, we didn't even have the first genome sequence. But even when the Genome Project was over, I thought, yeah, maybe in my life we'll see this, but I certainly won't be in my professional career. And I was once again wrong, and it does happen. I'm frequently wrong. And it's remarkable what's happening. Estimates of 25,000 in the US and maybe 50,000 patients worldwide every year are now getting their genome sequenced for suspected rare diseases. In some of these cases, they have a hint of what might be going on, but in other cases, the condition's completely undiagnosed, and that's, of course, led to this whole popping up of undiagnosed diseases programs. And in the setting of the neonate and NICUs, where now rapid genome sequencing is becoming mainstream in some settings. I'm hoping that those settings will be growing with time, where acutely ill newborns for which the neonatologist has practically given up on the child having no clue what's wrong with them. The idea that you could just sequence their genome, not only do it, but do it fast, and not 100% and probably not even 75%, but something like 40, 50, 60% of the time, figure out what is wrong with that child, and in many of those cases, be able to change clinical course and in ways that they hadn't anticipated, and in many cases save their lives. And you don't need very many of those examples to be convinced that the needle has shifted on this. Prenatal genomic testing, the onset of non-invasive prenatal testing is actually the #1 genomic test right now. Estimated 5 to 7 million pregnant women worldwide will get this test now done routinely. That number will likely grow. Part of the reason the significant uptake, there's a great industry for it, and I think part of the reason was a lot of obstetricians were already involved in this, and this was easier. It actually threatened some of their income, as you know, because they no longer have the procedure, but of course I think it's for the betterment of the pregnant individuals who can do a simple blood draw instead of a more invasive procedure. But this is remarkable, and it's all come about because of the new technologies. Without the new technologies that were exquisitely sensitive, we would not have seen this kind of uptake for non-invasive prenatal testing. And then, of course, pharmacogenomics, which we've always recognized that theoretically has great opportunity. I think still probably hasn't reached its peak by any stretch of the imagination. And there's a lot of barriers that all of you are probably familiar with getting practicing physicians to use this routinely. We need more evidence. I think maybe Josh Denny, who sort of works heavily in this space, may have some things to say about it. And I think the All of Us program that he'll describe may be one venue to push the evidence to a stronger position, so there'll be greater uptake. But still, there's plenty of good examples of this, and it certainly deserves being on this list. Lots and lots of eyes are on genomic risk and the idea of polygenic risk scores. And before I let this look up here like a highlight, I immediately have to animate and point out I put a question mark here. This is not mainstream. This is not real. This is research. Our institute is certainly among those doing lots of research now to try to see how polygenic risk scores might be relevant in clinical practice, and importantly, how we could make them be relevant in all populations, not just of those individuals of European descent. Once again, I think the All of Us program will be very influential here as their data comes in, giving great opportunities to maybe advance the use of prediction models using genomic information. I hope this list grows. I bet it will grow in the coming years, but I think nonetheless, we can look, especially at those first 4 areas, and just think about 20 years after the completion of the Human Genome Project. One sequence is all we had, no knowledge about really how all the complexities of the human genome. And here we have changed the practice of medicine in nooks and crannies, not across the board, not everything. And I'm not even including in here because it's not human genomic side of thing, all the infectious diagnostics that can take place because of advances in genomics, and that obviously became front and center during the COVID-19 pandemic. So those are the celebratory things, but genomics still faces big challenges. Every one of the things I'm about to say could probably take me a full hour to talk about. I just want to just list them for you. I want to make sure you appreciate that NHGRI is humble in appreciating that we have things that still need to be fixed, and these represent big areas for us. You know, sure, I say you could sequence a genome of a neonate, of an undiagnosed patient, and you can figure out what's wrong with them. Well, it's not that simple, right? I mean, really clinically understanding a genome sequence routinely remains very difficult. And what we're doing right now is cream skimming of what we understand, and there's a lot more that needs to be done, and making it facile for busy healthcare professionals, put it into their workflow in a fashion that they can truly utilize this. It represents a grand challenge that we are certainly fully embracing. You know, we also embrace the fact that, you know, we're no longer a little boutique field of genomics that maybe could just go off and sequence a human genome and not think about its consequences. It's just the opposite, because as genomics has become more mainstream in so many areas of everyday life in general, but in particular healthcare. I mean, oh my God, once you touch healthcare, what's more complicated for almost any society than healthcare? And now here we are touching healthcare, and that makes us very relevant for society. Obviously, that is why the Institute has embraced studying the ethical, legal, social implications of genomic advances from the beginning of the Institute, essentially. We continue to do so in a very robust way. But every time we turn around, we realize that, wow, we're no longer a little boutique scientific field. We are a field that's profoundly relevant to society. And that comes with immense amounts of responsibilities and attentiveness that we take very, very seriously. Among those serious things include things we have not done well. We have a significant diversity problem. We've written about this. We're funding various initiatives to try to fix this. It's multidimensional. Some of it relates to the fact that just historically, we have just not paid enough attention to our research participants, so that way too much of today's genomic data is from individuals of European descent. They don't represent the cross-section and full diversity of America, let alone the world. But I will in advance applaud what Josh is going to tell you, because all of us is part of the solution here. It's not the only solution, but it's a big part of it, where diversity has become a huge priority for them to help do a course correction on behalf of the whole biomedical research community. And I think we're also seeing similar things worldwide. But we can't ignore this with respect to research participants. The other aspect of it, of course, is our workforce. Too much of our workforce is not diverse, and especially, and there's so many reasons why this is so important. But all of you, I think, will appreciate, as individuals who think about healthcare applications, the medical community needs to look like the patients, and to get that comfort level and trust. And genomics is complicated, and I think we have to have a circumstance where our workforce has to reflect our patients, and it doesn't, and we need to fix that. And so, again, one of the big priority, or I'll talk about this a little bit more, a big priority area for the Institute is, can we help enhance the diversity of the genomic workforce? And I mean everything from scientists all the way to healthcare professionals of all types. And lastly, we also have to be humbled, and we are the Genomics Institute. We will always be the Genomics Institute, and our expertise will be genomics. And genomics is incredibly important in health and disease, but we also have to be humble and realize it's not in this alone. We have to also appreciate, especially when it comes to common diseases, there are both genomic and non-genomic factors, and ultimately, to fully understand these, we have to break down silos and recognize that we need to understand all these dimensions and how they interact. And so in order to fully appreciate all the underpinnings of human health and disease. So we celebrate. We're also face daunting challenges, big challenges, but that doesn't deter us. We're an incredibly optimistic community. And so I think many of you appreciate that starting back in about 2018, we began a new strategic planning process, thinking about the celebratory elements, thinking about the big challenges in front of us, and we got ideas and needs and proposals and obstacles, and we went around the country. We talked to this group, I remember, and I know we talked around the country, multiple places, and we collected a lot of ideas, lots of ideas, great ideas, and we synthesized those, and somehow we did our best by hearing lots of terrific proposals and nuances of what was going to be needed for the future of genomics, and condensed it into 10 pages in Nature, which is what we published in October of 2020. And the key aspect of this strategic vision, every one of our strategic visions has sort of a signature aspect to it. And this one was how, you know, once upon a time, NHGRI was all about genomics, and then maybe 10 years ago, we were mostly about genomics, and no longer can we represent all of genomics or even most of genomics. We can only represent the forefront of genomics, but only is a huge responsibility, and it's the really cool, fun, cutting edge, front ebb part of genomics. But we have to appreciate that, you know, here you are on the NIH campus, you know, every institute here funds genomics research. And in fact, now our institute funds less than 10% of human genomics research. Once upon a time, we used to fund 95% of human genomics research. The world has changed around us. That's a good thing. We've gotten out this discipline of genomics everywhere. But it means that when we're going to throw out a vision, we need to put out a vision that's at the forefront, because the rest of it is, we need far more of the scientific community strategically planning in their areas of how genomics is going to be used. That was 2 years ago, or now a little over 2 years ago. We got to implement. So that's what we're busy doing. And we can't implement everything we heard, because we heard all about genomics. We had to synthesize and define what was the forefront, but we are busy launching new projects and new programs, new initiatives, and new policies. So this is clearly a big part of what we are doing now. Let me just give you a little bit of a slice about what some of these things are. As I mentioned earlier, diversity is a huge priority for us. We made the decision that once the strategic vision was published, the number one thing we would get out of the gates doing is to focus on workforce diversity. In fact, we didn't wait for the paper to be published. We had secretly, not that secretly, within the Institute, we were already planning for what we were going to do in this area, since we knew what we were going to say in the strategic vision. So we got a running start. And so literally January after the strategic plan was published in October, by January, under the superb leadership of Vince Bonham, who's our deputy director, we put out a document that described building a diverse genomics workforce and how we have an action agenda for making this a priority area. And Vince and I published the very first paper in the American Journal of Human Genetics in 2021, and it was this perspective, and outlined a series of goals that we were going to achieve. Let me pause for a minute, and rather than go into significant details, I'm going to show you 2 slides about this. But just on Monday of this week, Vince Bonham gave an update about this, what's been happening with the action agenda. And it was a beautiful description, a summary. My video team will be posting that like we post all of our council meetings, the open session part of it. And so what I would tell you is that when that gets posted, and I don't know if it'll be later, they're very busy this week, but that'll be either this week or next week. When that goes up, I strongly urge you to watch Vince's presentation. And Donna, maybe if you could remember, when you see that go up, maybe you could send out to this group the exact link to Vince's presentation, because I couldn't do it justice. But it is worth knowing all the things that we are doing in the space. So he said it better, and you can see that video probably within the week. One of the things that Vince talked about, and we're very proud of, is a recognition that we needed, in order to get a better functioning in diversifying the workforce, we really needed a better structure in the Institute to align with this set of very ambitious goals. And so I created, and I don't create structural changes in the Institute very often, but I created last year a new office called the Training, Diversity, and Health Equity. Can't quite see it. Gerald, can I do this myself? What do I do? I say hide the floating meeting controls? Yeah. Okay, oh good. Oh, there you go. That's our new office. It's a really, Vince is the acting director. We will soon be recruiting for a permanent director. Its goals are to develop, support, and coordinate our programs that foster careers in genomics, all the training, diversity, and health equity components of that, advise the different parts of the Institute about genomics workforce diversity and health equity, and provide strategic programmatic leadership. They have been extremely functional in their first year. Again, Vince does a much better job at this, but in his presentation, but just as a highlight, we're recognizing it can't just be us. We need partners in academia. We need partners in the private sector. We've been talking to industry. We are hoping to have some major industry collaborations in this front. Maybe we'll be announcing later this year. We certainly, we also gave it out for the very first time at council meeting on Monday, a new award to 3 new awardees. They're called Diversity, Equity, Inclusion, Accessibility in the Genomics Workforce Award, to really start to honor champions in this area and to reward those who we believe are really moving the needle on diversifying the genomics workforce. And then there's a number of funding opportunities and that we have put out to really put our money where our mouth is. And I think these have been really important to start getting out of the gates on some of these things. But of course, it doesn't just end there in terms of things coming out of our strategic vision. In addition to that, you know, the action agenda was the first thing, and I don't even want to tell you about all these things, but needless to say, we're busy. And these new things or renewals of things or advances of things we'd already been doing really represent that full spectrum of activities from technology development to genome function, to understanding how variation influences function, to thinking about genomic medicine implementation activities, including polygenic risk score predictions and so forth, and thinking about diverse communities and the relevance of that in making sure we don't exacerbate health disparities. So lots and lots of things, and you could always follow the developments by tuning into our open sessions of council, because every 4 months we highlight what is the latest, and all of that is put up on YouTube. But there's a lot of stuff. I was given these few slides because we knew you'd be particularly interested in things related to the training of healthcare professionals, but there are these new funding awards that have gone out to a number of groups. I'm just going to sort of scroll through. Here's the first set, and here's the second, the additional set. So again, we are putting our money where our mouth is on these initiatives, and we regard these as part of a broader effort to think about how we are going to get the clinical workforce prepared, along with working with all of you, to see these genomic advances have their maximum input. More broadly, we have, even before the strategic vision was published, we had embraced the idea that we should be investing more in training. The history of this was I put together a task force of my council to go out and really look at our training, extramural in particular, training portfolio, and see where our investment was in training more broadly relative to other institutes. And I learned that we were below average, and you know, us genomics types, we don't like to do anything but at least be average relative to other institutes, maybe even above average. But I've made a serious commitment now to move our investment so that we are more in line with the rest of the NIH in terms of the % of our extramural dollars going towards training. And so you also, and some of this is for training with a diversity emphasis, and some of it is just broad training, thinking about how our workforce needs to grow in general. And so there are various, I'll just list them here, just various new initiatives we put into place to invest more heavily in training of the next generation of genomicists and genomic medicine practitioners. The other thing I just wanted to say a few things about is the outreach, because I think it aligns with some of the things you think about, is the outreach in our communications efforts and our education efforts. And we really have, I think, built up some significant, valuable assets in this arena. These all end up being collaborative efforts between our education group and our communications group. The communications group, so of course, they physically gets it out on the web. And one thing I can tell you that has gotten significant usage is this talking glossary of genomic and genetic terms. This started years ago. We did this. In fact, it was called for just genetic terms then. And we used the pandemic as an opportunity to have a little bit of extra time, and this would be the kind of thing we don't need to do in person. And so we did a deep redo, not only a structural redo, but also a critical look to make sure we had the most valuable terms. And Larry Brody and I and a few others spent dozens, I'm sure there are dozens of hours on Zoom calls enhancing and re-engineering this, including working with our graphics team. So if you haven't seen, by the way, if you haven't seen this, you really got to take a look because it's really remarkable. We have all incredible numbers of terms that are now modernized for a 2023 lens. Many of these come with beautiful illustrations from our graphics artists, including some with animations. And then we have audio recordings from NHGRI experts. So it truly is a talking glossary. You can listen to Larry and I on some definitions, but you can also listen to people like Francis Collins on other definitions. And it's just, it's remarkable. And what I'm always just so delighted by is if you just do a Google search on a term, pick a term, meiosis or something like that, more times than not, we're the top hit. And it's not like we're paying for that or doing anything. There's no fancy, business thing to get us to be the top hit. We're the top hit because lots of people are coming to the site. Millions and millions of viewers are coming to the site, and these are everything from professionals to grade school kids doing biology projects. So if you're not familiar with this, please become familiar because I think it's in really terrific shape. Similarly, my communications group has worked hard with various collaborators within the Institute to put out fact sheets to help make genomic literacy easier on individuals who are just trying to learn some fundamentals about genomics. And we're regularly putting out these fact sheets, and we're regularly looking for new topics. I personally found that, especially with lots of discussion around variation and pangenome and things like that that are becoming common words, I felt it was time to come up with a more informative explanation about human genomic variation. And so we just finished and just put up, I think last week or maybe the week before, a brand new fact sheet with lots of beautiful graphics that our graphics artists worked on human genomic variation. So again, if that's a topic that you think you're of interest, you really should take a look at this. We put a lot of hard work, again, drawing on the wonderful generosity of some of my staff who are willing to jump in and get involved in little mini projects, and I take advantage of picking people from all across the Institute to do this. And then the other aspect of my communications group is they have a history of genomics program embedded within our communications group. This sign I started a little over 10 years ago. I believe I'm the only institute that has on staff a doctoral level historian, which I have, and I'm proud of, and I have a whole program associated with this. And we're not only archiving a lot of stuff, we're also doing a lot of scholarly things and a lot of community engagement activities. And so now they're regularly putting on really important programs, all by Zoom, all recorded, all that you could watch. And here are just two examples of two that we've done recently, really tackling topics that are really difficult, challenging, that draw on historical situations, but also thinking about them in the current lens and just making sure we don't repeat problems of the past, needless to say. So again, I think this is a very scholarly, intellectual group that is really attractive. When we have these events, we're literally getting hundreds and hundreds of people joining us by Zoom, and then many, many hundreds of people watching these things on YouTube afterwards. So we feel we are doing something that is of broad interest to people. And we also have significant social media. My new communications director, that was the big thing she wanted to push when she joined me a few years ago. Big social media presence. I now have a very active Twitter account. So does the Institute, and we also do fun things like Ask Dr. Here's the one I did with Josh. Ask Dr. Genome, where we do a Q&A through Twitter about once every other month. One I did with Josh back in March, and then one I did with Karen Miga, talking about the telomere to telomere consortium and so forth a little more recently. And really, and this is my last slide actually, is continually thinking about how we can be helpful to people when they come to our website, genome.gov, and including providing a one landing page that if you go to, which is easy to remember, stay connected, genome.gov/stay connected, where I welcome you to this page, but really hope that you just scroll down and we just give you lots of places to jump in to either to my monthly newsletter, to my Twitter account, to our strategic vision, to our brochure, the talking glossary I told you about. On the bottom left is the workforce action agenda, and then lots of things in Genome TV on YouTube, and also from our history program, our oral history collection, which is growing and growing all the time. And then lots of other opportunities to either listen to me, either watch me or also to read some of my op-eds. So again, we try to make this as user-friendly as possible, and I would encourage you to not be shy. If there is some resource, or there's some fact sheet, or there's some definition you want to add, we love input from folks, either from you or from people you're talking to, say, oh, wouldn't there be a great, if only there was a place to go and learn about such and such. We'd love that input, and we're doing a lot of things constantly to collect that input. And so please don't be shy about letting us know when there's something we could develop that would be helpful to you or to the communities that you interact with. So I didn't think I fell a half hour, but I guess I did. But I don't think Josh would mind if we took a few questions before I turn this over to him. So let me stop there. And
I'll give, just to mention, I don't know if Donna mentioned this because I came in a bit late. There's almost 50 people tuning in remotely. So we're getting questions and discussion and connections going on in the chat.
Great. Okay.
So,
I hadn't appreciated who's remote, but
I'll just ask one question that came in from Tracy Weiler. There was before you even got to our efforts in training and the grant mechanisms and the new programs, there was a discussion going on about genomic education, especially in the medical, pre-medical space. It's one I know you and I talked about with respect to your son just recently.
Yep.
And so Tracy wants to know what are your thoughts about training for genetics and genomics literacy for the non-geneticist healthcare professionals? How does this fit with the strategic plan? And there was also an earlier question about genetics in the medical training programs.
Yeah, Larry, you're welcome to jump in. We talk about this a lot. First thing is I always try to say, I'm not meaning to be precise, I always try to say we really have to address issues for healthcare professionals. I try not to say physicians and doctors only, because I completely realize that's only a piece of the puzzle, and it's just as important that we think about pharmacists and nurses and the nurse practitioners and physician's assistants and et cetera, and certainly genetic counselors. This is it's like anything else. The truth is most patients don't spend most of their time talking to their doctors. They talk to other healthcare professionals because the physicians are just on the move, too busy, et cetera, et cetera. So it needs to be across the board. We want to know how we can help. That's why we're doing some of these modules and various other things, and we're looking for you to help tell us what's missing. And I would certainly say that, what I know the most about is medical school, because that's where I went. I didn't go to, and Larry mentioned about my son. I have a son who just finished second year of medical school at a terrific medical school, but I will just say that I was disappointed in how much time they devoted to genetics and genomics in the first 2 years of medical school. But I also know, and I'm sure Josh would echo this as well, that changing curriculums in medical schools is probably as complicated as getting everybody on Capitol Hill to get along. It's complicated sociological barriers to it, but we'll keep pushing, and we're totally receptive to ideas. And every place I go, when I visit, I try to meet with the dean and of the medical school, and I try to convince the dean that they need to pay attention to this, because this is going to become so important. And there's a lot of competition for time in front of medical students. Yes.
Hi, I'm Shamitha Zashkova. I'm the past president of APHMG, and we had just published a set of competencies for the undergraduate medical education field with respect to genetics and genomics. You're absolutely right that there's far too little time spent on genetics and genomics in the medical curriculum. I think the last time we looked, the average was somewhere around 35 hours in the whole first 2 years. And given the incredible advances in genomic sciences, that's fairly far too small. Also, given workforce issues, we really need people who are not specialists to understand these topics.
Yep.
So we're looking for different ways to partner with institutions to get these competencies out so that healthcare professionals are aware of the changing landscape of what would be the appropriate information for even non-geneticists to know. So would love to talk to you more about that, and also get any interested folks active in APHMG as well.
One of the things I think we would look to a group like this is if you think there's people we should be talking to. If you know where we can move the needle, you should really let us know. I think there's a lot of motivation. Here at our institute, I even think more broadly, because I know I can get Josh to come along. He would agree with all of this. But we just need to know who to talk to, or we think we can actually make things happen. So.
All right. I know the slides went by quickly. There's been questions about the slides. I'm sure we'll make them available.
Absolutely.
Certainly, there was a lot of URLs of funding programs to create modules, not just for the physicians, but for other healthcare professionals. We'll make sure all of that gets distributed to everyone who's registered for the meeting.
So maybe one more question, because I don't want to cut too far into Josh's time.
Is there, well, maybe we should move along.
Okay.
If that's okay.
Totally fine.
But
So,
thank you, Dr. Green. And
absolutely.
I think, you know, when there was that, the vision process, and it was great, you know, ISCC-PEG was involved, and we sent recommendations, and we definitely want to help continue with that, and we really appreciate you joining us today. Just a quick logistical thing. I don't know if Roseanne or Donna, there is a lot of good stuff going on evidently in the chat, but at least for me as an in-person, it's hard for me to find the link. I think, Donna, can we send the link to people in person as well? Or Roseanne, because I think I'd like to be able to look along what people are chatting and people be involved in that discussion. And it might be buried somewhere, but I had trouble finding it in my in my email. So, but someone should be able to have that. And right.
We can send out the registration links again. It's a registration, so you need to use your unique Zoom meeting link. But we can
so
send that.
So that's the issue, because we all registered for in-person. We never got that.
When you registered, you should have automatically got that as approved on a web page, but then you need to add that into your calendar. I'll send those out again.
If you don't mind, just because I got that probably a couple weeks ago, and it wasn't in my calendar.
And we're recording this? And also the chat will get captured, and so.
Yeah, but.
I understand, no, I know what you're saying. Real time would be more helpful.
Right, if people, and I see people have their computers, so.
Absolutely.
If they open them up, they can join in that as well. But thank you so much, and,
Okay. You can.
Is Dr. Green going to introduce?
Yeah, I'm going to introduce Josh.
Perfect.
Which I'm delighted to do, and Gerald can change over. So it's certainly my pleasure. I don't want to give a long introduction, although I have a lot of great things I could say about this wonderful individual. But Josh Denny is here. We finally dislodged him from Vanderbilt. It took a while. He was somebody who had gotten his undergraduate degree and then his medical degree, and then even got a master's degree in Vanderbilt, did his training there. And when all of us was being conceived, even before it was all of us, Josh, being a leader in this field, was somebody that NIH relied on heavily to be on the key architectural group that designed the All of Us program. And lots of arm twisting to get him to come here to help us run All of Us. The timing wasn't quite right at the time All of Us launched, but then a few years later, the timing was right. And so he's a very distinguished physician scientist, member of the National Academy of Medicine, the American Society for Clinical Investigation, and the American College of Medical Informatics. But he's also an incredibly nice person. We're practically neighbors. He just recently moved into my neighborhood. And he's also someone who's a real cutting-edge clinical researcher. And he's probably not going to talk about his own work, but maybe he'll touch on it. I don't think so. But he actually has his own research laboratory in NHGRI's intramural program. So not only is he a colleague of mine at the institute director level, because his position essentially is the equivalent of an institute director running the All of Us program, but he's also part of the NHGRI family as one of our intramural investigators. So All of Us program, which NHGRI loves and roots for and sees so many valuable connections to the field of genomics, and we were certainly heavily involved when the plan was first conceptualized, when President Obama got all behind it. And it is just in great hands with Josh Denny, who's now here as a local colleague, and I'm sure you're going to learn a lot about the All of Us program and what he's about to tell you. So Josh, thanks for coming.
Eric, thank you. Thank you so much, Eric. It is really a pleasure to be here. And, you know, I've had a long association with NHGRI and see familiar faces here as an awardee when I was on the other side in different networks, Emerge, Ignite, PGRN, and then of course in informatics spaces. And you know what you do as thinking about education in the space is, I think, very important as well. I got my start in informatics in medical education, actually building natural language processing based systems to develop and extract content from medical education content. And so we got all the lectures and document handouts, and we extracted concepts from them. And actually, one of the first things we actually did was to mine all this content, some of which, by the way, were manually typed documents, not PDFs, typed pieces of paper that we scanned and OCR'd for their teachers, and were able to give them back Microsoft Word copies. So one of the first applications of this, besides just delivering electronic content to the students, was creation actually of a genetics course. So we created a de novo genetics course by finding all the content in the curriculum, surveying and seeing where it was taught and how much it was taught, and then pulling together key faculty and creating a new course. So what you do about getting that message out and trying to get there is a very important message. And so today, that's not what I'm going to talk about, though. I'm going to talk about the All of Us research program, and it really builds on a lot of what Eric talked about both the advances and the opportunities we have in genetics, and diversity being a hallmark of that. And I'm guessing I'm using the mouse to move. How do I? There we go. All right. So I'm going to start a little bit historically. And so there's lots of cohort studies and things that have been built. I'll pick one, the Framingham Heart Study, and just think a little bit about how much we've learned from it and how much we have advanced science in an interventional way by learning about, and watching cohorts. And so a small number of people started in 1948. This is the first publication in 1961. So it took 13 years for that knowledge to be, derived and hit the medical literature. And, in this paper, we saw those curves that were sort of accepted as normal of having cholesterols of 300 or systolic blood pressures of 175 as just being part of the curve, just being part of that. And then seeing that, these do look like further evidence would confirm intervenable, risk factors for bad outcomes, cardiovascular disease, stroke, et cetera. And, about this simultaneous with this, you could actually, go back and look at, even the logs around Franklin Delano Roosevelt's, death and his physicians treating him and even, telling him to smoke to help lower his blood pressure and reduce his, anxiety. Of course, obviously not what we wanted to do. And so, what we have learned from this has dramatically provided and reduced outcomes. And you can look at similar curves for cancer, other diseases as well, where we've really been able to improve mortality or morbidity from disease from learning about intervenable risk factors and then finding treatments, of course, for those. And with things like All of Us, we think that we can close some of those gaps and make it relevant to everyone. So Eric talked about the lack of diversity in genomics. When I first made a slide around this, it's ironic because I think it was so this would have been in 2015, and we were using data maybe from 2009 or so, I guess. I can't remember when that first papers were published. But the interesting thing was the diversity actually, we talked about it then as a problem, and diversity got worse over time, not better. We didn't expect that as we went through 2015 and 2016, 17, that for a while we'd actually get worse with global GWAS diversity. But you can see where we are now. It's still over 90% European for these genome-wide association studies. And, you could go on, and probably you know this well, the impacts that has on real outcomes. And, that could be our polygenic risk scores, that interpretation of, pathogenicity, both from the standpoint of, maybe understanding pathogenicity in general within a given population, but also in terms of just knowing about the variation and observing the variation that occurs, of course, in diverse populations. And amongst those, 6,000 diseases that we now have genetic interpretations for, ones that may have ancestral differences, of course, will also have differences in understanding of them from that standpoint as well. And so that was from the beginning, a driving impetus for All of Us was to really sample more of diversity ancestrally. But, as we thought about the program in general and things we could put into the program, historically, we started with things, if you think about Framingham or other similar cohorts, survey instruments, structured exams, genomics, and then through efforts like eMERGE and other biobanks that started leveraging health information, capturing health records as part of that and useful for information. It's interesting, when we talked about this in 2015 for how to build all of us, EHRs are actually still very controversial as a source for genetic phenotypic information. But of course, it's become almost a standard across all major national cohorts now. EHR information derived in different ways in different countries is a hallmark really of all these cohorts now. Mobile technologies, other sensor-based technologies, I think is something that we think about today. We talked a lot about as a foundation, of course, so many other types of molecular information you could get at, metabolomic, proteomic. We've been talking more, I think, and more awareness of environmental issues, social determinants of health, which have environment and social structures to them, putting these kinds of things together. And as we've grown in the EHR space, we've been capturing radiology imaging, for actually a longer time with defined standards than we had around electronic health records. But that's another opportunity that we'll be thinking about over time as the kind of data, hopefully, that could go through the same sort of availability and reuse, repurposing for research as maybe we've seen with electronic health records. And of course, it's with all the buzz around AI and machine learning approaches. Of course, recently that's been in a textual standpoint around ChatGPT. But before that, we were really spending a lot more time thinking about transformative impacts that it's had around imaging and making that accessible in different kinds of ways and new technologies. And we're actually leveraging, some of those things. We hope to be leveraging some of our ancillary studies as well. So the mission of the All of Us research program is to accelerate health research and medical breakthroughs, enabling individualized prevention, treatment, and care for all of us. And it starts in these three components. And it starts with nurturing relationships with participants as partners that we expect to go on for decades, of at least 1 million participants who reflect the diversity of the United States. We decided from the outset that this was not going to try to be a representative cohort. We were going to try to over-represent diverse populations. And based on what we've seen in the communities, we also wanted participation to be open to everyone, but we wanted to build that relationship. We wanted to partner with participants and really engage diverse communities. We really wanted to focus on engagement. We want to deliver this research data set into as many hands as we could safely in a protected way that was secure. We want to think about the model for which we did that. And then we had to think about this in an ecosystem context of funders, researchers, participants, to really make this, as our goal, an indispensable part of the future of health research. And we have a series of code values that we established from the beginning that guide us. Participation's open to everyone, and that is within the U.S. It reflects the rich diversity of the United States. Participants are partners. I've talked about that. We'll talk about that some more. Transparency is key to earning trust. That we wanted participants to have access to the information through early learnings. We discovered that that access to information and things that we can augment that is a real return of value to participants and part of why they might be part of the program. We wanted the data to be broadly accessible to researchers, really highlighting security and privacy, and we wanted to broadly influence change in research. And so that's one of the reasons we do some things that are not necessarily directly within the All of Us research program's goals. For instance, like we help support ISCC, we're involved in GA4GH, a lot of these global communities that are promoting ways that we think of interactivity maybe nationally or internationally, helping get return of value out there. Think about this. We did some engagement with COVID, things like that. We want to be a positive catalyst for change in research in general. So we have a lot of participants, networks of participant engagement partners, community groups, medical centers, of course, doing the bulk of enrolling, and federally qualified health centers. The VA is part of this as well, and then technology partners in here. And so this gives you a survey of a lot of those different awardees. The bulk of our enrollment happens through what we call our HPO, or healthcare provider organizations. Those most of the slide is filled with those names. Though they recruit most of the enrollment from our program, and most of that's in person, but we also can, especially during COVID, had to establish ways to get out to participants virtually. And so now we've actually really can legitimately say we can reach anywhere in the U.S., including U.S. territories, through things like saliva kits that we can send out, through partnerships such as Quest, where there are 2,200 locations across the country that you could get a box and take it to a Quest location, and they could draw the necessary blood samples. And then new partners such as blood banks that we're working with. So we're really trying to expand our reach to enable ourselves to reach potentially anyone in the country. In fact, we actually can also, one of the newer partners, Exam One, that can go to a participant's home, and we're looking at to especially reach people that are can't necessarily travel in, and to be able to capture things like blood and urine that we can't necessarily get with a saliva kit if you're disabled, if you're elderly, or if you're just in an area we can't otherwise reach well. We have a number of community partners as well. We've, from the very beginning, had participants involved, or potential participants, in every stage of governance of the program, in our different committees, the steering committee, executive committee, providing voices back to us. And in fact, we even started with engagement studios in 17 different cities, 77 different individual engagement studios to reach diverse populations and hear, what might you think about a program like this? What should we call it? What is precision medicine? What would be valuable in this? What would you value in specifics in terms of getting information back? We launched nationally in 2018 in May. Since then, we're approaching 600,000 participants that have consented, 430,000 that have donated biospecimens. Our real focus on, I said participation's open to everyone. Our engagement efforts are, of course, focused mostly on underrepresented populations, and we think about that in a very broad sense, and you'll hear, I'll go through that in a second. As we sample towards collecting biospecimens enrollment, those efforts is where we, really think about how much your contribution is and where we invest our dollars, and especially in terms of engaging diverse populations. COVID hit us hard, like really any program. We were over 99% in person in terms of collection of biospecimens when COVID hit, and all that stopped on, March 18th of 2020. And so we had to figure out new ways of reaching participants, and that took a while. We also safely relaunched those individual facilities, and some of you have connections to those, to know how we did that with initially a lot of social distancing, sometimes using tents, different ways of going about and restarting the enrollment process. We are still the country's in a different place now, and we're reaching people differently. And that's affected different communities and different hospitals in different ways. And so some geographies are right back where they were in, pre-COVID. Some geographies are not quite in terms of the pace of enrollment. But overall, we're certainly approaching those kinds of levels again. We actually had our best overall recruitment numbers for December 2022, was our best month overall of any month we've had in terms of total participants that have joined. So we're really regaining, I think, some of that momentum we had before. And I've talked a lot about diversity, and so these are some of our numbers. We have aimed for, when we started, over 45%, over 45% of our population by race and ethnicity would be diverse, and we're exceeding that. And then we think we want over 75% representing underrepresented populations in the broader context. And you can see the ones we look at. We've grown this list actually over time. The most recent one we've added here was disability as an assessment, and these participants are part of our program. They belong to our program, and so we send it at some point we put that in the normal set of questionnaires that people did. But for the people who hadn't completed that, we sent them an update questionnaire. So it just gets to the we've had to do a number of things that we've done in an ongoing way with our participants, and so over 80% of our participants are underrepresented, given all the categories that we look at on the right. For most of these, we overrepresent compared to the U.S. population. Things like rural location, we don't quite, but we're pretty close. And then some of the subcategorizations of race and ethnicity, like Asian population, is under compared to what the U.S. population is. So we have funded, for instance, a network, the Asian Health Coalition, which has a number of partners to help us engage that population and help increase that. In terms of data that we collect, it's the same, despite how you come in, whether it's from a healthcare provider organization or whether it's a direct volunteer. You see the categories here. We have a website, we have an app. This is a screenshot of the app on my phone and my EHR information. Part of the reason to show this is, I actually enrolled as a direct volunteer. So I don't have a healthcare organization sharing my electronic health record information to the program on my behalf. So one of the things we did to allow people who enrolled as a direct volunteer like myself is, I can link in different providers through this interface. And so I've linked in Hopkins and Vanderbilt, where I used to live and where I get care now. And it puts that in an interface. I can see what that looks like. It has links out to things like Medline Plus, so you can read about the medications or conditions that might be in there. And then you can also see in the other kinds of information, you'll of course see things like, have you, your physical measurements are taken for the program. You can see your survey results, some compared to other people. Of course, if you donated by a specimen information. So we talked about returning value to participants. And so I don't just call this return of information on purpose, because there are many ways in which you can return value. And that's some of the things we heard. So in our engagement studios, as we've surveyed people, most of the people, in fact, early on, 91% of people said that, they would participate in the program regardless of what we gave them back because of, belief in the mission of the program and what we're trying to do. But, these kinds of things help people. We think it's kind of the right thing to do anyway, but it also is part of the process of being, sticky with the program, and we want to give that kind of value. And we think about this more holistically. Importantly, it's also part, I think, of how we affect change in how healthcare and precision medicine is adopted in this country. So what we heard strongly from our participants was the genetic information, especially genetic health information, would be the most valuable kind of content to get back. Not every community, as you might imagine, rates and values things the same way. And so you have to think about this, just like we talk about precision medicine, precision engagement, precision return of value. But overall, genetics, especially pharmacogenetics, actually was what was ranked highest, and then health-related other kinds of information. So in addition to the other stuff we return, you saw a little bit of the screenshot of some of the early kind of, I would say, pilot this front on EHR. We're robustly invested in return of genetic information. We started with genetic ancestry and trait results, and so you can see what those are over on this slide. And then that was December of 2020, we launched that. And then we launched genetic health-related information, and that launched in the fall of 2022. The first has gone out to, actually, I think it'll be on a future slide, certainly over 100,000 people. The latter, we did pilot, we went through an FDA approval process, and it's been launched. So I guess over 180,000 people now have been notified of genetic ancestry and trait information being available. You can see what this looks like in our interface. There's information to review it. We, of course, did a lot of testing with participants in the process. And you can see what people generally look at. Most people that say yes look at the ancestry information, and then a smaller number of those look at traits. And some people look at, of course, just traits or just ancestry, but you can see that the numbers are all pretty close of what people look at there.
Josh, do you mind taking a question?
No, we can take a question in the middle. That's fine.
It's coming up right now. A couple of people in the chat are wondering about healthcare providers' involvement in the data that's being returned to the participants.
The involvement of healthcare providers. Yeah, I'll work in that into my component as we talk about the health-related. So that's a great segue to talk about health-related return of results. So the two elements we return are based on the ACMG 59 right now, and then pharmacogenomics. This is an example of what the reports look like to participants, and we launched this starting in December 13th, that we announced it at least. Right now, we've notified nearly 17,000 people. The report is available to request. They go through a process. We don't require anyone to get results back, so they are notified that they can get this information if they want it. They go through a set of informing loops that describe what it would be like, what the potential risks are, and then if they choose to do it, then we generate the data. So then we call the variants based on pathogenicity, we call the pharmacogenetic variants. If it is a ACMG variant, we validate that with a second sample and orthogonal process, and then we give them their reports. And so right now, about 7,000 participants are approaching seeing their results, and we are working to scale this up. About 1,000 to 2,000 people get notified each week that they can request generation of their results. These are actually my results. I have spent a lot of my time researching pharmacogenomics, was one of my areas. I also worked some on ACMG kind of results and other things. But it's interesting, amongst pharmacogenomics, I, of course, spent the most time on CYP2C19 and clopidogrel. We built a program at Vanderbilt that did interventional prospective pharmacogenetic testing, including CYP2C19 and SLC01B1 and other variants. And it's interesting because I think in our current, at least in some of our platforms, we have not even included star 8. It's kind of interesting that star 8 is, of course, what I ended up having as one of the loss of function variants in CYP2C19, which is relevant to clopidogrel, a antiplatelet drug, and one of the more common antiplatelet drugs given. And then simvastatin being one of course, our anti-cholesterol medications and one of the most common out there as well. I think it's declining over time as other things have become generic that are powerful as well, but it becomes relevant to home, which is kind of fun. In terms of involvement of providers, we heard strongly from participants that they wanted these results to be returned to them and then have the actionability. The other thing that's relevant to this is within the FDA process to get an investigational device exemption, these become research results. So these results cannot be directly returned, for instance, within the electronic health record. And so but participants wanted it that way as well. We have both a provider-friendly handout and a sort of participant-friendly handout that you can be generated from the website to sort of facilitate that transition. In addition, genetic counselors are available for anyone that wants to request information and talk about their results. All the ACMG 59 results are delivered through genetic counselors at that first point of contact. And we have support for English and Spanish, and then over 200 languages through a language line for that return of information. So a number of our different HPOs are actually setting up genetic clinics or precision medicine kinds of clinics to handle this on their end. Pitt has one, I know, and Arizona has set up one. There are probably other things that are happening other places to handle that as well. I will also point out that for ACMG results, we do, when they talk to genetic counselor, we will pay for a clinical confirmation, and when they're talking to genetic counselor, can fire that off directly to the participant. So during that conversation, if they want to order that test kit, it's a saliva test kit sent to them, and then they can send that in, and they'll have a full. It's not so much CLIA that deals with, it's the FDA IDE that makes us a research result, but this will be a clinical test that they can use for actionability and guide clinical care. So far, everything that we know about is validated both on our internal secondary validation and the few cases I know about that have gone through the clinical confirmation as well. These are, we're running everything at a really high grade, and so I would expect that to be the case at this point. We also have a number of assets to more broadly support our different communities here. You can see some of them here to just talk about what it means to have clinical DNA testing, the results, and insurance. Some of this is, part of the process in educating what may be relevant participant, and when they learn about it, GINA and the limitations of GINA, these kinds of things that might be relevant as they learn about these results. We're, of course, listening to what we hear as we do this and things that we might need to learn in the process. And this also includes information for clinicians. They can also, just to point out with the genetic counselor, they can contact them again. If they have further questions, they could contact them with a provider if they needed to. That service just remains open to them. So we want to help support people through this process. Now we're going to shift and talk about researcher access. So we think about this in terms of tiers of access. I found with some of the work we did, for instance, at Vanderbilt in setting up a DNA biobank, that one of the things that was really helpful in accelerating research use was having tools that were easy to query and just to see whether or not you had a certain number of people with a given condition. And so one of the things that we launched, and the first thing we launched, was a public tier that was supported by what we call a data browser, where you can just get basic counts on diseases, drugs, labs, get some estimates of like the range of these kinds of things, and how old they were, what their sex at birth was, things like that, to get some basic information on across the different, what's there. And then we have tiers of individual row-level access to do the analysis that is behind, an application process. What we did here is developed a cloud-based model that was launched in May of 2020. The control tier is a more identified, or I shouldn't say more identified, everything has obvious personal identifiers removed, but it has more data in the control tier, including genomics and real dates. And so the idea is you should fit your project to the tier that it needs to have to do the science. So, if you're not using genomics and you don't need real dates, you should use the registered tier. It's exceeding safe harbor standards. For instance, if you need, you want to do something that is temporal in time, you need real dates, you would use the control tier or genomics. And, we try to guide you through that process as you set up your workspace. And then over time, we're going to have access to things like individual biospecimens, participant data, and these future ancillary studies. So this is what the data browser looks like. You don't need any login for it. Anyone can access it anywhere in the world. You can access it from your phone, and you can search in terms. What I like showing with this is it lets you walk across the different terminology standards, ICD-9, ICD-10, problem lists. There may be in SNOMED, different prescribing terminologies. They get mapped into a common backbone using OMOP if you're familiar with common data models. And so you put in cancer, you get a nice tiering of concepts from malignant neoplasm to disease to all sorts of other different individual cancers in the hierarchy, and you can explode those as the terminologies map across. I wanted to show you what it looked like to search for a gene, and so we just put some filters on this as well, so you can search for all the ClinVar variant interpretations. You can base that on other types of interpretations, allele frequency, those kinds of things. So there are different filters on top of this data. They're all fed by sequence data, and this just shows results for CFTR that we've observed with 244 variants in CFTR that are ClinVar P or LP. And if you click on those, by the way, you can get the breakdown by ancestry. For instance, thousands of people have P variants in APOL1, for instance, in our cohort, but given the diversity of it. So you see those kinds of ancestral differences for sure in those. So our research access through this thing that we call the Researcher Workbench is a cloud-based model. We wanted to bring people to the data instead of having the data be downloaded. Bring researchers to the data, the analytic compute, democratizing research access and process. When you come in, you actually authenticate the researcher with their institution, not the project is the design. So researchers basically get a passport to do projects, and then they can create new projects on the fly, which can be dynamically reviewed. And most of the institutions sign what's called a master agreement with us, which means that any new researcher that validates, usually with the institutional email address, can start access in about 2 hours from the time they first log into the resource, if that institution has a master agreement. It currently is open to U.S. nonprofit and academic and healthcare organizations. There's a few for-profit in that academic space that have also have access, and we are looking to expand that access as we speak, and hope to be able to announce more in the nearest time frame on that. The data, of course, that we curate and make available to researchers lags behind where we are with enrollment. So this is what's currently available. We'll have a new release coming fairly shortly in 2023. Total in that research component is about 374,000 participants. you can see that, relatively small for us, 12,000 have shared Fitbit data with us. These are people who just linked in their devices that they happen to own. We are also doing a pilot where we're giving out 20,000 Fitbits to especially diverse populations. But even that is actually a really big data set for publicly available data. And you'll actually see some of our highest profile publications right now have come out of the Fitbit data set. And until we had genomics, that was the most numerous of our data points, actually, because of course, the time series component. But even though we started nationally in 2018, we collect the extant data from things like EHRs and Fitbits. So the EHR data, as you can see, goes back up to 40 years in some participants. That means that even though we don't enroll kids yet, we have more than 20,000 virtual kids that have shared data, in some cases from birth, from their historical electronic health record data. And with our genomic data set, probably more of interest to a lot of this group here, we have about 100,000 whole genomes in the first set we released last year. It's about 165,000 arrays. We, of course, put in new cloud-based analysis there. In that data set, we observed 600 million loci with variation, and that corresponds with 400 million points that were not in Nomad, 400 million variants that were observed at least once, not in Nomad, 100 million variants observed in 3 or more participants, not in Nomad at the time. So of course we're working to share with Nomad as well, but it just gives you a sense of the kind of variation that we're seeing based on the diversity of our population. We're looking forward to a soon announcement. The next announcement will have over 200,000 whole genomes in it. As you look at our population, this is the diversity of that genomic data set compared to, in gray, the data on global GWAS data, which of course is mostly arrays, but it just gives you a sense of how we're really trying to seek and provide that data set available to researchers to diversify our genomic knowledge. Really the populations that represent African ancestry or Hispanic, Latino, or Spanish are incredibly low worldwide. And in addition to that, we capture the rest of the social determinants of health kind of information and other kinds of information that is multifactorial, of course, as we think about determinants of disease. And, amongst our population that self-identify as white, over 60% of them, have some other category in which they're underrepresented in most research. So we think that's an important way to think about the research story here. We have a goal for 2026 that we set nearer to when I started here at the NIH of getting 10,000 researchers on the platform by the end of 2026. That was based on, good available evidence and what other similar cohorts have done at a little bit more than, 5 years of launch and, sort of looking at about 7 years. You can tell we're going to beat that goal pretty handily, I think. We have approaching 4,500 researchers now at this time, and I believe it's over 4,000 active projects now. We're seeing the publications, pick up a lot, and that's what you expect. It takes a while for the momentum to build on publications in most of these kinds of resources. One of the things we're really trying to do is reach out to diverse researchers, just like we are participants. Just as Eric said, we need to have a research community, and we want to grow that biomedical workforce to look more and more like our participant population. That's important to our participants, but it's important for diversity of hypotheses to be explored, how we get these data implemented, those kinds of things. And so we're reaching out to HBCUs. We've partnered with MIHD. We've partnered with these NHGRI, some of these awards that Eric mentioned. We're a partner on, and we're have already given out supplements to some different researchers based on being part of different networks that can help us reach diverse researchers. And so we're actively thinking about this process. We also have, I like the nonprofit organization category as well, because we have things like patient advocacy groups in different diseases that are actually some of our, at least initially, were some of our biggest users of the platform coming in and doing research for their, patient community on given conditions, and, reporting that back, using that for, policy and how they direct their money, and also trying to stimulate, research and do their own research in that space, which I think is an exciting use case as well that we typically don't think about in the research community. So what's it look like to do a GWAS in type 2 diabetes? So I want to use this as an example. When I started in eMERGE, we initially selected 5 different phenotypes, and we had 5 sites that we're all going to explore. And one of those was type 2 diabetes, and it was one of the early ones that came out. It took us about 3 years to do that. We developed a phenotype at one site, we validated it, and then we spread it to the other sites. We each ran it within our sites. We pulled the data together in a federated fashion, so we all had to send it to each other. We had to curate it and clean it, and then do the analysis. And to do the analysis means having, the necessary compute infrastructure to do that analysis, which is not cheap. That algorithm is there on kind of that flow chart in the bottom left, because electronic health record data is not perfect. Now, at that time, we were doing things like manually writing misspellings for names of medications to search for, because we didn't have coded information. Now, in our resource, you can actually build a point-and-click algorithm that mimics this, and it does the terminology mapping for you across type 1 diabetes codes, type 2 diabetes codes, ICD-9, 10, all that kind of stuff. So this took us about 3 years across these 5 sites and a lot of funding to do the first time. In this process, I gave it to a postdoc in my lab, and I wasn't actually intending to like time him. I just told him, hey, let's just do this. I want to see how much it costs, how hard it is to do. Can you replicate this? And so he went through and he developed a cohort using the cohort builder. So it's a point and click to build out this algorithm. And then he dumped it into a data set, which is also point and click. And then you get there, after that, you get to a notebook. So you have to do R or Python to actually do the analysis. But you can develop that data set and dump it into a notebook. He adapted an existing notebook to do that, and that gives you the Manhattan plot in the bottom right, and nicely replicates the well-known signal TCL7L2. This was actually on this data set's even evolving, but he used 23 million SNPs in the process, about 20,000 participants, about 4,000 cases, I think, and the rest being controls. It took him about 20 minutes. He point and clicked and fired up about a couple hundred CPUs to do the analysis for him, and it cost $37. And the time I emailed him to the time I got this Manhattan was 3 days. So we have, it just kind of gives you a sense of how much more accessible this becomes. And by the way, he did this on his laptop. He didn't use any big, powerful compute infrastructures. He just leveraged ours and was able to kind of expand and do that. So we hope this really becomes the paradigm that makes this much more equitable across researchers to do this kind of research. And so we're even going, actually one of the Regeneron Science Fair winners in high school this past year used all of us. And so, we're hoping to continue to get that message out there to, other kinds of audience that wouldn't necessarily have these big, huge compute infrastructures with the right guidance to be able to do these kinds of analyses. So what kinds of diseases do we have in our resource? And this just gives you accounts of some things that we've done. Most of these are just generated with the cohort builder. We did some deeper analysis on COVID-19 here, and for the alcohol abuse, we used Audit C-scores as one of the many standardized resources we have in there. I already talked some about access to researchers and how this works. The key point here is we really want to facilitate access, and if your institution has a master agreement, that new trainee, that medical student, they can get started that same day and doing analysis. So I really want to encourage continued adoption there. What can we support? So, any large, broad kinds of association analyses across many things. We think this is, you have drugs, you have diseases, you have surveys, you have social determinants of health. We're building out resources around mental health. We have repeated surveys during COVID. that, you can look at changes in things like exercise and financial conditions and things like that as well. So all those things are available. One thing we don't do is we're not a representative U.S. sample. I've said that before, but we've doubled down on that. We don't have uniform follow-up on all our variables. We have far fewer Fitbits than we have electronic health record data, for instance, right? But we're still going to put in as many of those kinds of resources as we can. And we are not a real-time resource. We are pulling from things like FQHCs, which some of them literally started with Excel spreadsheets for their electronic health record. So they don't have real-time data feeds on their electronic health record. And so, we lag in production of our research data set. That's good for most use cases. It's not, where we want, it's not where, we were not the best resource to look at COVID and real-time responses on COVID. These are sampling a few studies that have come out. I'm going to dive into a few of those. These are the most impactful from 2022. You can see, I guess, the first one, the second one were both Fitbit data. Actually, the fifth one on this list was around wearable use in FQHCs and what their populations thought about it and what the barriers to use were. And then you have some that actually deal with mental health and all of them really, you have a theme more of diversity. So this is an analysis that looked at Fitbit data and the phenomic impact of this. So one of the things I worked on was doing so-called phenome-wide association studies, or PWAS, in electronic health record data. So this is mapping incident diagnoses based on step count, looking at a certain time, after that step count. And what you can see is walking more steps, of course, impacts a lot of diseases. That's not a surprise. But what's neat is you can quantify it, and you can look across the phenome there. And as a former primary care provider, I respect the fact that, first of all, it's not 10,000 necessarily for most diseases. And if I could just get someone to walk from 1,000 to 2,000, that makes a difference. 2,000 to 4,000, that makes a difference. You can look at the shape of the curves on the right and see that there is it's not necessarily linear. There's an effect though, and sometimes it's, more exponential of how many steps you walk and the impact on disease. And, it also, you don't have to get them to 20,000 too. We can kind of confirm that more with these kinds of data. This is a study looking at the so-called Latino epidemiological paradox, where it looks like people who identify as Hispanic Latino may have lower incidence of cardiovascular disease. In this case, in this analysis, these researchers found that didn't seem to be the case. It actually seemed to be a greater risk for most of the population. The other thing I like is the first author was a resident. So it just gets to that story again, that we can get trainees involved earlier doing this kind of work. And of course, it was led by diverse scientists as well. I mentioned mental health, and this is kind of a year where we'll have some larger investments in mental health. We are launching a mental health and well-being module that will be coming out shortly. We have an ancillary study that gets at quantitative aspects of things like cognition, risk-taking behaviors, things like that, that can feed into understandings of mental health diseases as we look forward. And these are research studies. It's based on brain games, or some, we're seeing lots of companies get into the space. We're developing a resource and testing it in a pilot phase now to look at how that goes, and then to roll it out in a large way, assuming the pilots go well. We have partnered with NHGRI on some workforce grants, and one of those in process now, I believe. We also, across a number of institutes and centers, have launched an R21 and R03 program, which we have 27 institute centers and offices signed on to. So one of the key things, I think, for all of us is success is to broadly serve the NIH mission, and the mission across all diseases, all stages of life, and resilience, healthy aging, these kinds of aspects. And so we want to do that, and we think these speak to the broad support and applicability of the resource. We are also continuing to always think about our technology and how we can meet participants better where they are through a new award, the Participant and Partner Services Center, and better filling in research gaps like the Center for Linkage Acquisition of Data to pull in environmental data, which we really don't have much of now. So that's an award that will be coming, and other kinds of data to fill in some of the gaps we have from things like electronic health record. Eventually I'd love to get more things like dental records and things like that. Well, we talked about imaging, continue to pull in information that participants are very willing to share with us. They have given us broad access. We have high buy-in on those kinds of things. And also returning information, including environmental data. So with that, I'll say thank you, and I'm happy to take some questions for any time that we have.
Siddharth Prakash from UT Houston. I have a question about returning reclassification of variant data. As you know, a lot of us struggle with that, even with our small cohorts. What infrastructure do you have in place for reclassification?
So we have 3 genome centers that look at validating these results. And as they do that process and call the variants, it's synchronized within the system, a common system. And if it's been classified before, they'll see what the classification is, of course, as they go through the process. If they reclassify that, we would actually then know everyone who had that variant and what their reclassification status would be. These are going to be rare. We don't actually expect that reclassification necessarily to happen that often, but we will know it. And then the idea would be over periods of time that we'd actually look more comprehensively through the list to see if anything has been reclassified. More likely, it's probably going to be moving from VUS to something than sort of reclassifying down. But we don't have a firm protocol for that in place. We're certainly thinking about it, and the common data infrastructure should help drive it.
So I'll just thank you so much for speaking. I'm Rich Haspel. I don't know if you're here. I'm one of the chairs. No, you should stay up here.
Okay.
I just wanted to help guide the questions. Can people probably hear me if I speak from here? Yeah, that'll work? Okay. Oh, okay. So I didn't see, so yeah, are there some other questions from in the room or? Yep, go ahead.
Hi, Emily Edelman, the Jackson Laboratory. Thank you so much for such a wonderful update. Really exciting on a number of fronts. My question is about the release of results to the participants and how all of us is thinking about preparing the clinical workforce for receiving these results.
This harkens a little bit back to my days at Vanderbilt when we were trying to deliver results to participants and, well, really through the clinical infrastructure. And I will, Ebony's in the room, who was very part of a lot of the Ignite efforts, but as was Phil and probably some of the rest of you. But I would largely say I felt like my efforts trying to get that disseminated more outside of Vanderbilt underwhelmed what I wanted it to achieve. And so one of the goals for this program is by handing it to the hands of the participants who want these information data, that they, as they take it to providers, it will help facilitate that kind of change. We have provider-friendly handouts out there. We have the resources, and then most of these participants will be coming through HPOs. And so the HPOs themselves are often thinking about how they're going to receive these information, what's going to happen. Now, we're having to think more about what happens to FQHCs, for instance. They have less of the deep bench to think about this, and there are thoughts underway on that, but I don't know exactly what's going to happen, what we do in that space, but we're certainly thinking about it within that context.
No, I think there were some other questions on the chat, sort of, in some way. I mean, this is amazing. I mean, the amount of data you're getting. But I happen to be a pathologist, so we talk a lot about pretest probability and testing people who don't have, necessarily risk. So you're releasing a lot of data. So the question is about, one question I have is there plans for follow-up of how these people did that? And because, our group is all about education, is there an opportunity, either with that or some other way, like you mentioned, there were handouts created. what was the group that created, we're a big fan of sort of education science and really thinking. So my main question is, what sort of opportunities do you see maybe for educators? Are there opportunities in all of us for us to get involved and maybe help out? So
I like that question. Phil, I'm going to ping you, since you were so involved in pharmacogenomics worksheets and things like this. Is there any idea of like making these more public? Because a lot of the recommendations, and I mean, especially in the ACMG side, it gives things like percentages and stuff like this. And I'm thinking about the education piece first, and I can answer the pretest probability part second.
Yeah, I think you covered the handoffs back to HPOs and some groups thinking about doing that, and there are available things online to sort of, I don't want to say evangelize, but explain, the utility of the results. But I think it's an interesting question in general, because I think the education piece of it, we all know in this group that there's a gap to fill on the provider side in terms of education as well. That I think it's a great question of how we can simulate that.
It's a good idea. We'd certainly, one of the things, if you think about the pretest probability question, that's why we're sort of conservative on which results, we're sending out there. So the ACMG gives us an external body to say, we think the penetrance is high enough based on enough evidence out there that these warrant return in this non-selected population for this condition. So, but yeah, one of the things that's really important is getting the data back from these, calls into the resource to find out what happens downstream from that, right? What are the findings? What are the extra tests? And so, just like everything else, our EHR data will continue to follow them. We'll continue to ask them survey questions. We have not yet developed anything specifically around following up for this, but we might do that as well. And that evidence base needs to be generated to accelerate science.
So I think Dr. Ostrander.
Yeah, hi, it's Bob Ostrander. I'm the American Academy of Family Physicians representative to this group. And I practice in a village of 600 people in the second smallest county in New York that accounts for 0.1% of the population of the state. I often say we're from East Ohio and not New York. And it struck me, the underrepresentation of rural populations and rural healthcare delivery systems over and over is a concern of mine, in a place where I've made a lot of effort, and I kind of caught the footnote at the end. The one area we're worse than the U.S. population is with rural health. And my two questions are, is this because people in rural areas are not recognized as one of those groups that needs an effort for outreach, or is it because we're so hard to reach because of the situation of poor internet culture, and the fact that with all the consolidation of healthcare systems, a lot of our rural areas have been pulled into big healthcare systems that are urban-centric, and they don't really care about us either?
I won't comment on what other healthcare centers think or not think. From the standpoint of us as a program, we looked at our networks that came in on a variety of factors, and rural outreach was from the beginning actually one of those factors we looked at. And then we just kind of watched the numbers and got numbers, and we were pretty happy that we actually were hitting targets on most things. And then of course you're not going to hit all of them. And I think that is one of the areas that has been underrepresented. I would always say, this is an opportunity. If you have ideas and you wanted to talk more, something AAFP or others would potentially want to engage in, we can now reach anywhere, through things like saliva kits. So at the minimum, if not other approaches. So yeah, those new approaches are new for us, by the way, and they were developed during COVID. But of course, one of the benefits that we saw as obvious really was being able to reach everywhere and be able to better reach those who are in rural populations. So internet may still continue to be a barrier, but for the most part, I think we should be able to overcome that.
All right, I think, all right, one more, we can take one more question, but if you could come up and, oh, sorry, Larry, did you? Two more? Two more, okay, great. 3 quick ones, 2 long ones, and 5, no, just kidding. Let's just, okay, yeah. So who's first? Why don't you ask your, yeah.
Heidi Ongst from the Institute for Genome Sciences at the University of Maryland. What is the status of the pediatric program, and I had heard that it would be up and launched soon, pre-COVID, we had heard that. I was at Cincinnati Children's prior to this. And if it does get launched, how will you change the return of results? Who will it go to, and which will you change the genes you'll return?
Well, for that second question, how many weeks do we have to talk? For the first question, so I joined in January 24th, 2020, just to give you the NIH, to give you a sense of my timeline here. Obviously, a few things were just beginning to happen then. We did not have, let me just say it, I'll try to say it a different way. Let me be more proactive. There was a slide I happened to have in here that ended up, wasn't actually in this deck, but that actually got a little bit to this. We are really firmly back on looking and developing everything for pediatrics at this point. Those timelines got changed by COVID, for sure, like many things did, as we had to shore up our base. But as we hired our first director of pediatrics in October, we are developing that. We are planning to submit to the IRB within a couple months for that first infrastructure. We developed the plans of what it looks like, and we hope to be making more announcement about that soon. So it's one of my top 3 priorities for this year is developing the pediatrics infrastructure. So return results will change. There's lots of thoughts. One tidbit that I'll give you an insight into is that we're thinking about this within the context, especially for the younger populations, of having a parent guardian who also enrolls. So, so you have duos or trios, and in this kinds of situations, some of these results actually to return, and the implications for things like, biologically related, parents become easier to think about because they'll be an all of us participant too.
And Larry, is there a couple from chat? Okay. Yeah. Oh, could you put on do you have your microphone on? Oh. Oh, you want me to pull it up here? Oh, or.
Hit chat, I guess?
Yeah, I guess hit chat.
I can read Serene's question.
All right, yeah, why don't you do it? Thank you, yeah.
All right. Serene Hadar at St. Jude is asking, Josh, you mentioned that genetic counselors speak to patients about their pharmacogenomic test results. Do you have pharmacists as part of this team? And if yes, what's the role of pharmacists?
We have pharmacists to help develop, of course, content. We do not have pharmacists that are part of the return information at this point. Of course, the education of the genetic counselors and the materials and stuff like that are have been developed by multidisciplinary teams. So the pharmacogenetic results are returned directly, and so far, I'll just say, roughly 96% of people across all ancestries, but of course different for different ancestries, have actionable pharmacogenetic results if they are, exposed to medication of interest.
And was there one more? Oh, okay. So I just want to thank you so much for coming. I'm not a researcher, but I mean, it's amazing what's available. I'm going to share it with people who do research. And I just went to that single browser, you mentioned, and I have an interest in cancer genomics, and I looked up BRCA1, and there are 26,000 variants, like on 2,000 pages. So this is a tremendous amount of data, but that's why I also, we had some questions from, about pharmacologist involvement, pharmacist involvement, and just, we are a very diverse group with a lot of different interests, and we're very interested in education. So if you can keep us in mind, because I think there's personally, I mean, I'm just learning about it, and please, people should go to the website. I've already just looked through it a little bit. It's really, I found my institution, so I know we can get data, but people should check this out, but also let us know how we can help, because I do think there's a lot of opportunities here for education. A lot of data is going out there, and we're very strong believers in sort of very structured, evidence-based approaches to trying to get things out there. But this was least for me, this was incredibly eye-opening. So thank you very much for coming, and I just have to give it other.
Thank you.
That was terrific. So thank you very much. So I think what we're going to do, if okay with Donna, is we'll take maybe 10 minutes. Like, we just don't want to go too far over, but we want people to have a little break. So maybe we'll come back at 11:20, and then we'll get started with the scholar stuff. Okay? So thanks a lot. And thanks for all the people contributing to the chat as well. Sorry, we're trying to figure out how to integrate, but we'll get it done a little better. So thanks a lot.